Welcome to Unchatted Waters. In this episode, we talk about biodiversity in a changing world. From what contributes to defining biodiversity, to the past, present and future approaches used to discover new species. So if you're curious about evolution, food webs or even the tree of life, and how we untangle these complex topics, then this is the episode for you. Welcome to Unchatted Waters, the podcast that talks to the people behind the science. I'm Kirsty, your host, and I'm excited to introduce my two guests for today's episode. Peter Barry is a marine benthic taxonomist, identifying marine animals present at the seabed, including invasive species in the UK. Murray Thompson is a biodiversity and food web specialist. He uses data sets to understand who eats who and the interactions of species in our oceans. Welcome to the podcast, both.
Thanks. Thank you. It's nice to be here.
It's great to have you. So my first question is for you, Murray. What is biodiversity?
The way I like to think about biodiversity is a collection of species, and Charles Darwin described it really nicely when he described a tangled bank. So lots of different species interacting with one another. You have plants and their root structures that might go into a river, fish living in amongst it, and they're all interacting. Now, biodiversity, which is kind of a way of measuring species, or species richness, or or change in the composition of species, is actually...
Sorry, the jargon alarm's gone off. What is species richness?
So species richness is it's like the sum or the number of species in an area at a given time. Now, you might think that's really nice and easy to measure, like temperature. You put in a probe and you get a reading. But species richness isn't that simple because your samples don't give you a complete count of what's there. So often you have to develop statistics to test what's there, given how hard you're looking. So the harder you look, often the more you find. And you have to account for that when you're assessing biodiversity.
So is that because we don't have enough resources to sample the entire area? Because the world's pretty big, right?
Exactly, yeah, and species composition changes along gradients of environmental gradients. So let's say as salinity changes, you might get different organisms that can exist along that gradient.
So Peter, Murray has mentioned species. How do we actually classify a species? If I was to find an animal and bring it to your lab, what would you do with it?
Well, if you were to bring us something, first of all, I'd put it under the microscope. A lot of people, they bring me photographs of animals and they say, what's this? And I always say I can't put a name on it until I get it under the microscope because we have to manipulate the animal. We have to turn it over. We have to check different features on it. We'll look at the shape of different parts of it. We'll look at the colour. We'll often ask them, where did it come from? What depth? Even the time of year is important. And then we'll look at our literature. We have an idea of what should be out there. If somebody brings me, say, a crab, I'll start narrowing it down. I'll look in the crustacean literature, narrow it down to crabs. What type of crab? How many legs does it have? What shape's the carapace? And gradually, it's like a game of Guess Who. You just narrow it down and down and down until you get to something sensible.
I love the idea of playing Guess Who in the lab, trying to look at the different features and popping down That's a really nice analogy.
That's how it works.
Who's who?
That's my job.
Brilliant. So once you've looked at animals that it might fall under, is there a way that is described across, I guess, taxonomy? That was in your biography. Can you define taxonomy and how all the animals kind of group together a little bit more?
If you mention the tree of life, going back to what Murray was saying about Darwin, he started coming up with phylogenetic trees. Everything is related.
Sorry, the jargon has gone off. What's a phylogenetic tree?
What's
phylogeny?
Phylogeny, it's
a tricky one.
It's a tricky one. Phylogeny is basically how all the animals are evolved, how they're related, how they evolved from each other, from a common ancestor. And if you imagine phylogeny as a branch, at the base of the branch, you've got a common ancestor, and then as the branch goes up, you get little offshoots as different animals evolve. But they all basically come from the same thing, so we say they're related. Someone brings you an animal and you want to identify it, you go back to a basic thick branch, crabs, and then you start getting narrower and narrower. And you use the morphology, you use the shape of different parts of the crab that you're seeing under your microscope to try and find which offshoot does it belong to. And then you get it in finer detail. And then sometimes it comes down to, I'd say looking at the number of spines under the eye lobe or something like that. That's the finest detail, but that's what you have to do to get an answer. And then you can reliably say, I know what species I'm working with.
And it could be a new one, a new branch.
Sometimes when it doesn't match what's in the literature, you have to face the possibility that it's a new species, if nothing matches. And then you have to go and dig out an expert in that group, because you can't have knowledge of all the groups. There's just too much out there.
Sounds like a really interesting job where you're kind of a detective trying to find different clues and different parts of the story. It sounds really interesting.
That's the fun part of the job, definitely, yes.
One thing that we hear in the media quite a lot is that biodiversity is changing. So how, as humans, are we making it change? Where is this change coming from, Mary?
So a lot of the work that I've been involved in, we look at things like climate change and other stresses like fishing, nutrient pollution. There are some really key ones that we focus on, particularly in the marine environment. So in terms of climate change and fishing, actually, quite often they have quite a similar impact, certainly on fish assemblage. So with climate change, it tends to favour smaller organisms. Warming favours smaller things. And that's within species, so within a species they tend to get smaller, but also across species, so we tend to get smaller organisms towards the equator. And that's changing as the climate warms up. In terms of fishing, in a slightly different way, but it has this effect on fish where it's size selective, so fishing gear removes big organisms.
So one follow-up question I have for that is, why are warmer climates or increased temperatures, why are animals that live there smaller?
So it's to do with your fractal network, how your body is set up.
Fractal network. What's that?
So it's to do with how you generate energy and move it around your body.
Like metabolism?
Exactly. As you're making energy, it's more efficient in warmer conditions to have a larger surface area. So as it gets cooler, it's more efficient to actually have energy, store energy in certain ways. So
right, like blubber and fat seals and stuff like that, whereas you've got smaller animals at the equator. Okay. So apart from climate and fishing, are there any other ways that species and biodiversity is changing that we're contributing to? I think this is for you, Peter.
A lot of people think of it as a threat. It's not necessarily. It does get an awful lot of bad press, is invasive species. Human activities moving animals around the world. The world has evolved with animals in certain environments. They are where they are around the world because that's where they evolved, and sometimes there's biogeographic barriers keeping them in place. Usually an aquatic animal off the coast of America can't make it all the way across the Atlantic to British shores. So these animals, they just evolved in different parts of the world. Now you put in humans and human activities, and we don't mean to do it, but as an unintended consequence of ships crossing the Atlantic, going back and forth, you might pick up an animal and bring it across a huge distance that it previously couldn't get across. Sometimes this is animals clinging onto boats. Sometimes this is ballast water. Ships, when they're getting ready for a long journey, they pick up a lot of ballast water. They fill their tanks. You don't know what's going into the tank. You just open a valve, let it all wash in. Sometimes animals sitting in the ballast water tanks can survive the trip across the Atlantic. As boats have gotten faster, we've shortened the time they arrive. They're in a new environment. Nothing's ever evolved with them. Predators haven't evolved to take care of them, or they might have an advantage that the animals here have never seen. Introduced species, if it can establish here, start growing, it might put pressure on local resources. It might start taking other species' food. It might start occupying space that they would occupy, like oysters. There's some very fast-growing oysters that arrive. They like to clump together. They make huge reefs, and it just disrupts the whole environment. It disrupts water flow. They're all feeding. They all start taking other animals' food out of the water column. Sometimes it can have a positive effect. We generally think of them as bad, but there are some positives. For oysters, you could eat them. It's a positive for one species, which is humans.
So understanding those changes in biodiversity is really important in terms of what we're transporting across the world. But I guess we've been transporting invasive species since ships, so in wooden hulls of historical ships as well.
Yes, but the problem is that ships have gotten better, that you've narrowed the journey time across the Atlantic. If you think of going across, when Columbus went across, it took him, I don't know, maybe a month. I don't know if animals could hang on, if they could survive the extremes of temperature, of changing salinity in that time. Now that boats are doing it in a couple of days, you've got some tough animals that might just survive it. And that toughness, they have a high environmental tolerance. They can take the different salinities, they can take the different temperatures. The tough ones are going to survive. And when they get here, they get into a nice, relaxed harbour and they just think, oh, this is easy. I can outcompete everyone because, I survived the trip across the Atlantic. There is regulation, there is biosecurity, but it can't be perfect all the time. So some things are going to escape and establish and start to spread. And that puts pressure on the local environment. And it generally will reduce biodiversity.
It's a natural process, species invasions. In some ways you want it, right? It's because with something like climate change or these environmental changes that we see, species invasion is a natural process that will keep things going. So actually it's a useful trait in some cases. It's where we take organisms from one part of the world to somewhere completely new, and they do so well that they actually reduce biodiversity somewhere that becomes really problematic. So it's not always bad, but it can have really negative consequences.
Okay, so we've established some of the impacts that change biodiversity caused by humans. So why is biodiversity so important?
So the one that really gets me is, or I like to think about with biodiversity being important, is just simply the air we breathe, right? So some of it's produced by trees, but also a lot of it's produced by the algae in the oceans. So you get this combination of oxygen that will come from lots of different life forms all over the world, which I think is really quite magical. Another one that comes to mind is pollination. So this is about species interactions that produces food that we eat. That's really key. So there's lots of species interacting to produce things that we need. Another really important aspect of biodiversity that is how it affects society, right? So we have some really interesting migrations of people that track fish migrations, for example. So up and down the English and Scottish coasts, they used to follow the herring fishery. There's other fish migrations that people track. This actually has really large consequences on land, things that are happening at sea. So some of the fish that we eat in the sea, things like herring, mackerel, they're really key food for other fish as well. So we share prey with other top predators that might be mammals and seals and puffins. So there's some really interesting things that we act like just another predator in the system. So going back to Charles Darwin's Tangled Bank, then if you have lots of these species that are interacting, quite often it's good to have more complicated food webs, as it were, with lots of different species interacting. So if something happens to one species, the impact on that species gets kind of buffered around the whole food web. Whereas if it's a very simple food chain, you take out one, the whole thing collapses. If you have a much more complicated food chain, you can take out one species and then energy finds another way through the system.
Okay.
So having more species can be really beneficial to humans for having stable ecosystems that we can rely upon.
So biodiversity really sounds important. So I want to dive into food webs a little bit more and understand, how do we actually work out how this complex system works? Where do we even start with all these kind of different interactions?
So this is what excites me, which is slightly absurd, but this is how I cut my teeth as an ecologist. This is how I sort of began things, was actually looking at who eats who. So that actually often boils down to looking at stomach contents. So I started looking at stomach contents data in Iceland, in some Icelandic streams, which are heated to different temperatures by geothermal activity. And what that meant was, what it boils down to was me looking at midge larvae and dissecting them under the microscope to look at what they'd eaten. And we could find diatoms in their stomachs, so we can actually start making links.
Sorry, jargon map's gone off. What's a diatom?
So a diatom is a microscopic algae, and it has a silica case, and they're kind of beautiful, like glass under the microscope. And you can identify them by these different cases.
Even in the stomach? So they're
Even
not
in
broken
the stomach.
down?
They're half chewed. But you get enough of them to identify them. And then you would take some samples from the top of stones in the stream, and you'd take the samples of the insects in the stream and the invertebrates in the stream, and you'd look at the fish in the stream, and you look at stomach contents of the things that eat other things, and then you can actually make this big network of who's eating who.
So once you've observed those species interactions in the lab with your stomach contents, how do you think about that in terms of the other factors like space and time of year and seasons? How does that all then, if you've got, I'm imagining being back at school, and I know that we kind of drew out who eats who and drew an arrow to who ate the previous one. And I'm hoping that everybody else can visualise this as I describe it quite badly. But what other parameters then do we put on top of that, or layers do we add to that map once we've got that simple kind of arrow diagram?
This is a really tough challenge, and we try to tackle it in several ways. So it's really hard just to observe the species in an ecosystem, all the species, let alone all the things that they eat. You can never, I don't think, actually feasibly get to a total understanding of who eats who. But you can start to aggregate things, like if a fish behaves in a similar way to another fish, then you can classify them as similar feeding guilds. If they're eating similar prey, you can group prey in the same way. So you can aggregate these things and understand big bulk flows of energy. So that's how that's one way of doing it. Another way is actually looking as best you can at all who eats who, in as much detail as possible, to build up these networks, and then seeing how these networks change as you move through space or time. So that's what we did in Iceland. We were looking at how temperature affected network structure and flows. We do that in the sea as well. So there's an awful lot of stomach contents data collected from the sea. And that contains everything from tiny fish larvae all the way up to basking sharks. You can look at a herring, for example, which eats a lot of plankton. So they have these biased behaviours, as it were, these different foraging strategies. And they can tell you, looking at different fish, tell you different things about the ecosystem. So a herring might eat lots of plankton, so you can use them to observe plankton, whereas a plaice or a flatfish will tell you something a bit more about what's happening on the sea floor. And quite often you'll have fish at the top that eat all of these different things, and they tell you about the whole ecosystem. So these kind of the lions and the tigers of the sea, as it were, they can tell you a lot about what's going on just by, if you can see what they've eaten.
That's pretty cool.
Brilliant.
Yeah. So I have another follow-up question for you. If you've got these food webs, and I think what you've described there in the stream is quite static, if you've got a fish that is in a food web in the UK, for example, and then it migrates out of UK waters and is present in another food web, how do you work out the importance of that species in? Is it global collaboration that you will kind of share that knowledge, or how? Because fish move, right? How do you?
Exactly, we've had to do a lot of that, working internationally and collaboratively, bring together as much knowledge as possible about who eats who and about how we can sensibly categorise different types of predators and prey. That's still ongoing, and we do that by it's not just the species that behave differently, that eat different things, but it's also body size that drives a lot of these patterns.
Smaller fish are eaten by bigger fish, basically.
Exactly, so a small cod's going to behave just more like a small herring than it is a big cod.
Right, okay.
So because they're gape limited, their mouth, that's really the ultimate what they can or can't eat, right? So when you're small, you can't eat a big fish. I mean, it's just physically impossible. Body size is really important, and then species as well, because you have these evolutionary traits. Fish forage in a particular way, or all these organisms forage in a particular way. The ones that make my job easy are the ones that ingest things whole, because then I can see what they've eaten. But the ones that chew things can make my life really difficult.
Yeah, I bet. So once we have all this information, is there a way that we can test how different changes in temperature? So once you've got all that data, can you then change your temperature, maybe using computer models to understand how those animals may react to change?
Exactly, yeah. So we can do that in several ways. You can look at it on the individual level, so what happens to temperature to individuals, and that's really important because it speeds up things like attack rates or individuals' behaviour, how quickly they move, how quickly they burn energy. But then you can also look at what's known as an emergent property, right? Which biodiversity is one of those things. It's when you stack everything up, what does this quantity that's generated? So species richness being one thing, but also this might be a food web property like the network structure. And you can look at that into the future under different climate change scenarios and see how that might change. So if you can predict it well currently based on the past data, we try and use that information then to predict the future.
Starting from the beginning, you are mapping out who eats who, then you're working out how temperature and environment might change, how geography might change those interactions, and then you're putting it into a computer model to understand how things might change in the future.
Exactly.
Real nice.
And there's lots of different models along the way that do that, that
Yeah.
do different parts of that. And some of them share information and some of them don't. So, and often what you want is that they all give you similar or complementary answers. When they start diverging, you know there's an issue. This is why actually science is really exciting, because you have lots of different approaches, and when they all start giving you good answers in the same direction, you know you're onto something strong.
Brilliant. So Murray's described theirs from start to finish, and I thought I'd just ask you, Peter, how taxonomy has changed and is developing to forecast things for the future, or how I can imagine taxonomy started with people drawing species they saw along the same time period as Darwin and things like that. So how has it changed, like Murray's computer models? What kind of technology are we using?
Well, I would start off by saying a lot of the old descriptions that we use, they're really frustrating because they're about a paragraph long. I read one description for a little bivalve. The person who described it said it's small and it's white. And you think, great, thanks. A lot of these pictures they used were, they didn't draw it themselves. They gave it to artists who embellished it and gave it a little moustache or something. Or often we have what is called a type animal. With the first person who described it, they designated this is the one and only animal. I'll put it in a museum, and then every time you want to check, do you have the same thing? You come to the museum and you have to check it. We have the definitive article here. And that's called the type specimen. And a lot of times you look at the type specimen and then you look at this paragraph written in Latin and the silly watercolour painting of it, and you just can't match it up. So nowadays, when you're describing an animal, you have to go to the nth degree. You have to have brilliant photos of it, preferably photos of the animal underwater with all its little appendages out, especially for the colourful nudibranchs. They like photos of it underwater when it's happy.
Sorry, dragon alarm's gone off there. What's a nudibranch?
A nudibranch is like a shellless mollusk. It's like a little sea slug.
Nudibranch.
Nudibranch.
Great name.
It means naked gill. It's the gill that's on the outside of the... They're really cool. They're really bright sea slugs, like you say. They've got little gills on the back, and it's like, ooh, like a little flower on the back, and that's the naked gill. So
that's the thing about them. They are
no shell.
They've got no shell. So any fish that comes around could think, wow, tasty little treat. I don't have to crunch through that. So they've evolved an awful lot of toxins and stingy cells and just a warning stripe saying, lay off, you don't want to eat me. So they're very, very colourful. For a modern taxonomic paper, you're going to have just oodles and oodles of data on the morphology, because someone will come along later and they're going to try and split it. It's what we do. Everyone wants to find a new species. Someone will come along and say the fifth leg on this is slightly different according to the description here. So you have to describe all the parts in really fine detail. Thankfully, we can do that with the smaller species because we've got good microscopes. We've got really good light microscopes now. We can take really good photos down the microscope. B, we've got the scanning electron microscope, which arrived in the 1970s and revolutionized everything. I know for my own work, I was working on a bivalve.
Sorry, I didn't do the dragon alarm before. What's a bivalve?
Bivalve is a type of mollusk. It's got
a shell.
Sorry.
Because mollusk is also setting the alarm off.
Just think of a seashell. Two halves, like what you get in a restaurant. Two halves. And you open it up. And to make sure that they interlock again, the animal opens it and closes it and opens it and closes it to make sure it goes back in the right place. They've got little teeth that mesh together like gear teeth. That's really important for bivalve taxonomy. There was a shell that I was working on and nobody noticed that it had a little row of teeth, tiny, tiny little teeth. You could only see it under the electron microscope. And once I discovered that, I realized that we were looking at a totally different species to what everyone was calling it. And I said, hang on guys, this has a little tiny detail that no one's noticed. I think it's different to what we have been calling it forever. And it turns out that yes, it was a new species.
So I guess the reason people really want to find a new species is because you get to name it after yourself. Is that true? Or is that just a rumour?
You can't name it after yourself.
No.
No. It would be struck down straight away. You have to send your papers out for
a mollusk
review.
called Peter Barry. Peter Barry.
No, but you can name it after someone else. You can name it after your friends, colleagues, family. It's a great honour if somebody names after you. You've made it when a friend names a species after you.
Okay.
But you can't name it after yourself. That's
a no-go.
That's frowned upon.
So did you discover a new species then? Is that what you're saying with those extra teeth?
Yeah, with those extra teeth, and it's just because the advances in technology let us do that. We can throw everything at it nowadays. Just imagine the people who described this species 200 years ago, if they had the tools, if they even saw the tools that we were using, they'd be absolutely amazed. Having said that, we're not discovering new species all the time. The British Isles is pretty well documented, so it is exciting when you definitively find a new species.
How does it feel to find a new species? Guess when you find one, you're like, I've done it. I'm now in that list of people who's found a species. It must be pretty fulfilling. What does it feel like?
At the start, you're not sure because
you don't believe it.
Yeah, you don't believe it because there's so much variation out there. You have to check with experts. You have to go and check and double check. That's what makes our job difficult. You have to talk to all the people who know this inside out. You have to go to the museum.
Convince them all.
There's not a eureka moment. You have a little inkling. You think, I think I might have done it. And then someone will shoot you down and say, no, I've seen that before. It's not a new species.
Can I ask a question about is there a convention around coming up with a name for a new species?
No, you can name it whatever you want. I saw a species that was named, the Latin name for it was Thunderstruck because I like AC/DC and the song Thunderstruck. Yeah. There's another one, Dalek because it was named for a little fossil that looked like a Dalek from Doctor Who.
Oh. Was one named after Kermit the Frog recently as well?
Possibly, I don't know.
Oh, this is going to be a different Google search when we finish this episode of finding the great names. What did you name your species?
Similus, because it was similar to something else. I was in a hurry.
Don't like AC/DC or any other band.
There was your chance to really stamp your...
Oh, I said I'll have plenty of other opportunities. None have come around.
Ever confident. So are there any kind of other approaches apart from finding those physical, visual differences?
The traditional way of doing taxonomy is called morphological taxonomy. That's putting it under a microscope, taking a look at the shape or the morphology of the animal. What's beginning to happen now is molecular taxonomy. You look at the DNA and how they've changed. Going back to the start, if you look at your phylogenetic tree, it's how the branches have formed over time, how animals have kind of spun off. If you get populations that get isolated, they might just evolve on their own. They're not going back into the gene pool. Their genes will evolve different from everyone else. And they'll evolve different enough that you can say, yes, we've got a new species here. The genes are different enough. Revolutionising what we do.
So do you kind of backtrack through the DNA to see where that kind of splitting might have happened to create that kind of unique DNA signature?
Yes, you do it. We use computer models to make up a phylogenetic tree. The computer will tell you where it joins onto the tree, what branch it joins onto.
So I guess the computer outputs that both of you are working on kind of probably look quite similar because you've got these different species, you've got lots of lines going in different directions and the way things kind of fit together, I guess.
Well, so all of my work is completely dependent on what Peter does.
Yes.
So all of the information on species and who it's who, it has to be identified beforehand.
It's the tip of the iceberg putting the label on it. So make sure you put the right label on it. Because when you identify an animal, you get a lot of information with it. You get the whole backstory that goes with it. Yeah. You know what it is, where it should be found, what it eats. You need to put the right label on it. That's what I tell people. That's why taxonomy is so
Imagine just
important.
mixing up the labels on one day you have a bad day in the lab.
That's the one thing I've really stressed in field work to people is that write clearly on the bottles because it can cause so much issues down the line.
Okay, so we've talked a little bit about what biodiversity is, and we've talked about food webs, we've talked about defining species, we've had a really nice chat about how we name species, and now I would just want to ask you, Marie, what can be done to make sure that we can conserve and recover biodiversity? Because we described some of the impacts which is causing biodiversity to change.
The first thing with biodiversity is to try and unpack what biodiversity means. And we touched on this a little bit earlier, but there's lots of different ways of measuring biodiversity. So it might be the change in the number of organisms that you find across space and time. But it can also be things like change in body sizes and species and species interactions. So understanding which part of that biodiversity you're interested in trying to conserve or recover is the first major challenge. And we're making big developments there. And you can just by changing the space or spatial scale of your assessment, the hotspot of biodiversity, so where you might find particularly high biodiversity in one area, they move around. So this is why it's so important to actually understand what you're trying to measure. So once you have all those you've measured the different components of biodiversity, you can kind of stack them up on top of each other to find where across all these different dimensions, as it were, of biodiversity, they're all typically high. So then you can say, well, in this area, this is really high biodiversity across lots of different aspects. So this might be an area where you might want to invest particularly in conservation or protection. We work internationally all the time on these problems because organisms do not respect our national boundaries. So we need to work collaboratively with scientists, and also you work with such a broad range of people with different technical skills at that scale, which means you come up with vastly better solutions.
Okay, it's time to move on to the feature for our episode today, which is our fun fact. So this is a nugget of information or something we can take to a party as a conversational opener, something that we've learnt on this episode. So Peter, I want to come to you first for your fun fact, please.
The fun fact is we're still finding things. After 250 years of naturalists in Britain going down to the seashore and just hoovering up everything there and identifying it, there's still stuff left to be found, and not microscopic organisms. A few years ago, the scientist came in and brought us a little sea slug and said, what's this? It's a little bit strange. So I looked at all the appendages and I couldn't match it up to anything in the books. I went through all the literature, spoke to some experts, and eventually found someone who said, that's not right. That doesn't match anything.
They didn't shoot you down.
Didn't shoot me down, yeah. It was a nice feeling. But it just goes to show there's still stuff out there to be found. Still new species out there.
Wow, that's such a nice image that you're still finding stuff, but there's still more to be discovered. And I think that's really something that, as scientists, we love, that there's still more to find. There's still more answers to be found. So that's really great. We'll add a little picture of the sea slug onto the infographic for this episode so you can find out more about it. So it's over to you now, Mary, for your fun fact, please.
So I'm going to make it a little bit gory here. And if we go into the guts of the matter and start looking at fish stomach contents, if you find a big fish and you look inside its stomach, you might find that it's actually got observations in there of most of the ecosystem. So it might have eaten another large fish that will have eaten a slightly smaller fish that's eaten something from the seabed. And that something from the seabed has eaten some algae that's fallen from up in the water coming above it. So just by using stomach contents, you can learn an awful lot about the marine ecosystem.
Is there any way that you can microscopically or molecularly understand what's happening in the stomach if there's something you can't see with your eyes?
That's happening a lot currently, and it's a really interesting part of work. Because things like jellyfish are really hard to observe in stomach contents because they're digested very quickly and they just look like mush. So it's very hard to actually identify them. So molecular techniques give you information on who eats who at a much better level. It doesn't give you all the information. With molecular techniques, it's much better at telling you who eats who, but not how much.
Brilliant. Well, I really enjoyed this episode. I've digested a lot of information from both of you, and I really appreciate your time today. It's been really fascinating, and I can't wait to learn more about biodiversity in a changing world. So thank you.
Thank you.
Thank you very much.
Thanks. Thank you for listening. This podcast was made by CFAS, the Centre for Environment, Fisheries and Aquaculture Science. To keep up to date with us, follow us on social media using @CEFASGovUK. We hope you can join us again next time.